Hello, good evening or good afternoon. We are Luis Antonio, Emilia, Jose Manuel, Alberto Alejandro, and Jaime. And in this video, we're going to talk about artificial chromosomes. Well, we know about some already existing artificial chromosomes. The main types are the following. We know about the yeast artificial chromosome that are linear molecules of DNA that have elements from yeast chromosome, and these yeast parts allow the YAC to function as such. With them, cloning fragments, we can clone fragments of up to 500,000 kilobase pairs. Here we have a picture of, well, an example of like the structure of a yeast artificial chromosome. We have here the telomeric part. We have two because it's linear, so it has one at each end. We have the centromeric sequence, which is here colored as orange, the selectable marker TRP1 and URA3, the origin of replication, here marked as ARS, but it's also known as ORI. The restriction sites for cloning, which are here in purple. Then we go to the BACs, or bacterial artificial chromosomes. They are based on the F plasmid of 7 kilobase pairs, which is obtained from Escherichia coli, and can hold inserts of up to 300 kilobase pairs, though the average insert is 100 kilobase pairs. As any other vector, it has the ORI, origin of replication, multiple cloning sites, and selectable markers. That's like as we saw in class, the biobricks of any vector, but they also have the factor F. Here we have an example of the plasmid. In here we can see that it's of the vector that it's circular and not linear. Then we go to the hacks, which personally I found them very interesting. These are the human artificial chromosomes. They are not as widely used as the bacterial artificial chromosomes or the yeast artificial chromosomes, but these ones can be as long as 10 million base pairs, and they are basically a tiny chromosome that can transmit complete genes. It's quite useful if our gene of interest is especially large. Well, this allows the clonation of huge fragments of DNA, as I already said, and can be transferred to human cells for gene therapy. It's composed of a centromere, which is here in the image, obtained from a chromosome, of an actual chromosome. Telomeres, the same as in yeast chromosomes, which protect or are like caps that protect our vector, and our gene of interest, which is here, like with arrows, and that's it. Finally, we have the PACs, or the P1 artificial chromosomes. The P1 is a phage, so this vector is basically, it's also, you can find it also as phage P1 artificial chromosome. So because they are derived from this bacteriophage P1 and can carry inserts of up to 150 kilobase pairs. They require in vitro packaging, and Escherichia coli is also commonly used as host cells. This is an example of a PAC vector, which is plasmid JC PAC MAM1, and is 23 kilobase pairs long. So for this specific PAC, we have the P1 single copy replication. We have also the copy replication for low copy expression, and the multiple copying lytic replication on the black repressor for high copy expression in Escherichia coli. That's it. Thank you very much.